Question number 171. DNA polymerase used during PCR reaction is isolated from. And we all know that the DNA polymerase which is used in PCR reaction is Taq DNA polymerase. It's a thermostable enzyme and it is isolated from Thermus aquaticus. This DNA polymerase can survive high temperatures. So the correct answer for this question is option number 2, that is Thermus aquaticus. Let's discuss other options also. Very first option given is Bacillus thuringiensis. And this is a bacteria from which Bt toxin has been isolated. So this is an incorrect option. The third option given is Agrobacterium tumefaciens. It is a bacteria which naturally infects the dicot plant, from which Ti plasmid has been isolated and gets modified to form disarmed Ti plasmid, which is used now as a cloning vector. So this cannot be a correct answer. The last option is Haemophilus influenza, and this is also an incorrect option. So the correct answer for this question is option number 2. Let's proceed to the next question. Question number 172. Downstream processing involves. Now let's discuss first that what happens in biosynthetic stage. In biosynthetic stage, there's formation of products. So products are formed at this biosynthetic stage and then they were isolated from host cells. So after these products are formed at this biosynthetic stage, this is followed by the separation of these products from the reaction mixture, then its purification of the desired product, followed by its preservation. And these processes of separation, purification and preservation is referred to as downstream processing. Now let's discuss the options. The very first option given is obtaining the foreign gene products from host cell but not storage and selling, and this is an incorrect option because this is the step which is involved in biosynthetic stage and not in downstream processing. The next option given is culturing host cell in bioreactors, and this is also an incorrect option because it is a step of biosynthetic stage. The next option is amplification of gene of interest, and this is also an incorrect option because amplification of product also occurs during biosynthetic stage. The next option given is purification and separation of product, and this is the correct answer to our question because downstream processing involves separation, purification and preservation of product. So the correct answer is option number 4. Let's discuss our next question. Question number 173. Which of the following varieties of crop was patented by rice stack, which faced international outrage over allegations of biopiracy? Now the hint given for you is that it is patented by rice stack. So you have to select a variety of rice among the given options, and the correct answer for this question is basmati. There are nearly about 27 varieties of this type of rice in India, and it is this variety of basmati rice which was patented by this rice stack company, and it has faced the international outrage over allegations of piracy. Now let's discuss other options also. First option Larmaroso, second Sharbati Sonora, and third Atlas 66 are the incorrect options because these are the varieties of wheat and not rice. So the correct answer for this question is option number 4. Let's discuss our next question. Question number 174. Sparger in a bioreactor facilitates. Now we know that sparger in a bioreactor ensures oxygen availability throughout the reaction mixture, and the purification of these products occurs after biosynthetic stage. So spargin in a bioreactor facilitates purification of product. This is incorrect because this step is involved in downstream processing. The second option is maintenance of temperature and pH control. This is also incorrect. We have separate temperature as well as pH control systems in this bioreactor. The third option given is availability of oxygen throughout the process is a correct answer to our question. And the last option given is maintenance of anaerobic conditions, and this is an incorrect option because it facilitates the maintenance of aerobic conditions throughout the reaction mixture. So the correct answer is option number 3. Let's discuss our next question. Question number 175. In a continuous culture method, cells are maintained in which phase to ensure higher biomass? This graphical representation is for the bacterial cell growth, where initially the bacterial culture is in lag phase, which is followed by a log phase or an exponential phase. This is log phase. And it is during this phase the bacteria are growing exponentially. And in this continuous culture method, cells are maintained in this phase to ensure higher biomass. Because this is followed by stationary phase where the number of dividing cells is equivalent to number of dying cells. So we don't need this phase. And this is followed by a death phase where the number of dying cells are more than the number of multiplying cells. So among the given options, the very first option, lag phase, is not a correct answer because at this stage the cells are not growing exponentially. The biomass of the cells is really low. This lag phase is followed by log phase, which is a correct answer to our question. It is this phase at which higher biomass can be obtained. The next option is declining phase, which is the death phase, so this cannot be a correct answer. And last option is stationary phase, and this is an incorrect option. So the correct answer for this question is option number 2. Let's discuss our next question.